Good morning, everyone. Welcome back to the conference. Let us session 5. Let me introduce you to the chair of the current session, Professor Aditya Murthy. He is a neurobiologist by training and is currently a professor at the Center for Neuroscience at the Indian Institute of Science. His research interests span across fields of visual perception, decision making, and the generation of motor behavior. So now, without wasting any more time, I invite Professor Aditya Murthy as the chair.
Without further ado, I'll start the fifth session where the essential theme is Parkinson's disease research. And it, I guess, presents an interesting contrast to the Alzheimer's disease, which is probably a little less specific in terms of its progression and genesis. Parkinson's disease. Although Parkinson's disease is associated with motor disorders and motor symptoms, it's now a pretty well-established fact that there are huge cognitive symptoms associated with it, given the known anatomy, the close correspondence between the basal ganglia and the frontal cortex. And so I guess this would be the dominant theme of this session. Without further ado, it's a pleasure of mine to introduce the first speaker, Stanley Phan. Professor Phan is a physician, a very physician at Columbia University, where he was the founder and emeritus director of the Center for Parkinson's Disease and the Motor Disorder. He's also the H. Houston Merit Professor of Neurology at Columbia University, and among his numerous sort of work and research, he is a leading authority on Parkinson's disease. He's been modifying the gold standard, the UPDRS scale, as well as the Schwab England ADL score, which is used to determine the severity of Parkinson's disease worldwide. He's been involved in clinical studies, which have been actually very foundational in the research in this area. He's received his BS at the University of California, Berkeley, his MD at the University of California, San Francisco, his residency at the Neurological Institute at Columbia University. So without further ado, Dr. Phan.
I've been in times. I enjoy being in your country. I think science is great. I think the neurologists who I've met are terrific. So it's a pleasure to be back. It's particularly a pleasure to speak at the first inaugural conference of the Center for Brain Research. This is an amazing organization that's developing. Going to take off and do good things. We're looking forward to it, and this is the sort of the inaugural session for any of that. I thank the students who met me at the airport, chaperoned me and sent me in, and will take me back. They're very helpful. I want to thank them again publicly. I want to thank the organizer, and especially VG, not only a terrific organizer in getting all the but she got a visa for me. I was afraid to take it. And she personally entered the Indian Embassy in Washington, D.C. Just before Mali and say it's going to come, and I'll get it, and there I am. So thank you, VG, publicly for everything. So I want to talk about recent events in Parkinson's disease. In fact, I'm going to explain that the research in Parkinson's has been exponential in time, and you'll see in my slide what I mean by that. But it's really only 8 years ago that the first gene for Parkinson's disease was discovered. In this period of time of 8 years, we've now got new understandings of pathogenesis, new ideas how the disease progresses, and perhaps ideas of how to slow the progression. Most patients will tell you when they see them, they just have the first symptom. They have Parkinson's. Sure, they want to get a cure, prevent it, and go away and all that. But they will all say that if it will just stay where it is, I can live with this. But the disease progresses. And how do we stop that progression? We have a handle on it yet. We have some ideas and new ideas which I'm going to present in this talk. Some of these will work. Okay, so let's begin. Start at the beginning. James Parkinson almost 200 years ago, a little over a year from now, it'll be 200 years, 17, that he described 6 cases, 3 of them, and the others he just saw walking illustrations. You know, they have the flex, the elbows flexed, the bent knees, and they have the gait, and you can see Parkinson's. When you go to the market, you see people with Parkinson's. It's not difficult. He tremor, of course, and he described the tremor, and that was one of the things he pointed out was rest tremor. In fact, what James Parkinson astute, and those few cases he mentioned almost all the motor symptoms we know about today. And you see, I'm not going to read, you all know these anyway. He also commented, though, that the that's being injured, and we know today, maybe because we're alive longer, it's not clear why we do it, but 80% of people with Parkinson's disease will get dementia. In fact, my colleague Richard Mayu of Columbia said, other than having the gene for Alzheimer's disease, Parkinson's is the biggest risk factor for getting dementia. Of course, the kind of the dementia, mostly Lewy body dementia. Of course, also from aging and other things, get Alzheimer's on top of that or instead of that. Sleep does become disturbed. That's another non-motor disease. REM sleep, I'll mention that again, is pretty common. And constipation was mentioned. All these things were mentioned by James Parkinson. And as our chairman said, we now have a lot of non-motor symptoms of Parkinson's disease. In fact, that's part of what progresses. Non-motor symptoms are there before the motor symptoms of Parkinson's. After, all of these motor symptoms progress and non-motor symptoms progress. The new ones develop. And the worst one is dementia. And that's what people want. 1817 was when the disease was first described. Parkinson. It's possible to look in the literatures, the text in India and China. People try to point out maybe this is what Parkinson was, but it's not so clear-cut. At least James Parkinson was clear-cut. He called it shaking palsy or paralysis agitated in Latin, and that was the name that was stuck until Charcot later on, about 50 years later, it should be called Parkinson's because it's tremor and it's certainly not paralyzed. Paralysis agitans was not a good name. But it took almost 100 years to even know where the pathology was. So in 1817, it took almost 100 years, and we now know the pathology to be in, the predominant pathology to be in the substantia nigra, as you can see, the pigmented nigra, the normal one is the substantia nigra, and the Lewy body, intraplasmic eosinophilic inclusion, and it is a containing neuron in the nigra. Now, this, as I said, was 100 years. It was Fritz Levy in Germany who described this in 12, and in Paris, a Russian medical student trained in Paris, Konstantin Tritiak, for his MD thesis, brains of the post-symptom of Parkinson, it came out just about this time. He showed degeneration of the nigra. It was the first nigra being involved. In fact, he saw the Lewy body, named it the Lewy body. Levy, because it was a German, it was Levy. By the way, he changed his name through the anti-Semitism. The Nazis took his lab from him and demoted him and got the wisdom to leave before he was killed. Went to England and then America. He became a professor of pathology at the University of Pennsylvania. His cemetery is just buried just outside the city. Anyway, he changed his name to an anglicized name, Frederick Louis. So he wanted us to call it Louis. So we are Louis, which is the anglicized name anyway. But Louis didn't see the substantia nigra. He didn't even after Tritiak's paper, he wasn't sure about the substantia nigra being involved. He saw the Lewy body in the dorsal motor nucleus of the substantia nominata in the medulla. And there was this paper celebrating Louis' evolution, and that came out just a month after the anniversary year. And in that picture, we see this is the lab of Alzheimer's. He was trained in pathology. After his neurology training, he went into pathology, Alzheimer's lab. This is Alzheimer's. So we have a connection with, we talked about this yesterday, today we were talking about Parkinson's, and here's our connecting here. So he worked in this lab, made his discovery, he was recognized for that, he got his eventually, and that was when he lost the lab, the Nazis took in Germany. We're going to move 100 years. The next major event was the discovery of the dopamine story in Parkinson's disease. That took about 40 years from, this is a picture from 57, and 140 years after James Parkinson's. And this is the famous picture by Arvid Carlsson. In this particular picture, he took rats, resized them, and injected L-Dopa. And you can see the awakening. I'll come back to this picture in a minute. What was the story about it? Okay, so first of all, the story for dopamine really being in India. Rauwolfia serpentina is a plant here in India, and the Indian pharmacologist extracts of this and recognized how good it was in controlling blood pressure. It was the only drug at that time, when it came out, it was the only successful drug to high blood pressure. It also caused sedation, and what we know is what was called the sedating effect, we now know is Parkinsonism. Switzerland isolated the most active ingredient from our reserpine. Of course, they marketed it. That became a very drug in the history of pharmacology. The interest in how does reserpine work came to the Bernard Bode, who ran the cardiac pharmacology lab at NIH. Important people trained with him, and then one of his first technicians, Axelrod, who went at night to study PhD in pharmacology at Georgetown University in Washington, D.C., and he went on and eventually got his own lab, metabolism of the catecholamines, won the Nobel Prize. There were famous postdocs, Alfred Pletcher from Roche in Switzerland, in Roche Laboratories. He worked there, a postdoc, went back to Roche Labs. He became the research for Hoffmann La Roche. And there was Arvid Carlsson. Arvid Carlsson finished his doctoral degree in pharmacology in Sweden, advised by some mentor, you ought to go to Bernard lab at NIH. So he spent a few months there. By the time he got Bernard Bode had already finished, and they just published this work about reserpine's effect on serotonin in the brain. It depleted serotonin in the brain. Bernard Bode, that was the answer. That explained how reserpine works. It depleted catecholamines. Carlsson, in the meantime, when he was put to work, Carlsson was given to work on, I think, the adrenal medulla, and that works in that and complete serotonin. But Carlsson was very interested in couldn't look, let's help him look at the catecholamines. And Bernard Bode didn't want to bother. He said, no, we know the answer. That's it. But Carlsson was still intrigued. And when he went back to Sweden and he got his own laboratory, he worked with some other people, but then he decided to do that experiment with the rabbits. He did research, they flaked out. He injected the precursor for serotonin, 5-tryptophan, along with the decarboxylase inhibitor. Then he injects levodopa, and you see it. And I love this picture because the ears just perk right up and rabbits. Now, Carlsson submitted a picture with this paper, and the paper was accepted right away. There was no question about the paper, but they decided we don't need the picture. This picture was never published. And the Prize Award, in which he published it in his Nobel Prize lecture. He allowed me to use it, and and so forth. So the story isn't there. There are other things. His laboratory mapped out the regional distribution, showed high levels of dopamine in this striatum. Carlson had a more sensitive technique to measure dopamine in the brain. It's been measured in the brain, but it wasn't as a highly selective technique. Carlson developed showed that if you use resurfacing, you can deplete dopamine in the brain. You give dopamine in the brain, and recognize that it probably related to Parkinson's. But he did not do measurements in the human brain disease. It was Ornikavich who did that. Finished his MD at University of Vienna, and a postdoc with Hermann Blaschko in Berlin, worked on dopamine of all things to see how it was. And when he was finished with his postdoc, published, Ornikavich was intrigued. He used a method to measure dopamine. He got the brains in Parkinson's disease, post-dystopic Parkinson's, depletion of dopamine. That was the December 1960. It was immediately recognized around the world. It was paper, and whereas meantime, I should all this time was not thought to be but an intermediate in the synthesis of it was not thought to be a neurotransmitter its own. Carlson proved by many people that it could be. He showed the high depletion was resurfacing, and that gave him the idea. But it took many more years before it was accepted. Meantime, Ornikavich published this, teamed up with Berkmeyer, the natural experiment, to give L-DOPA. They developed L-DOPA, got some from labs. It wasn't Roche, it was another lab. I think it was Baron. Injected some, and found some awake periods. Unfortunately, really by other people, there are many studies following that. Some double-blind trials, some oral, some intravenous, and there was a mixed results, but the double-blind trials failed to benefit. And even as late as 1995, at a symposium in New York that the reason I'm even in Parkinson's disease, one was there talking his work and saying, well, L-DOPA looked to be a therapeutic agent. You have to realize it can only, it causes a lot of nausea and vomiting. Can't really give doses, and so it was doomed to fail. And then came along George Katsias, and George Katsias, he thought Parkinson's, he knew about the brain story. Dopamine had nothing to do with Parkinson's disease, but the loss of neuromelanin was the cause of Parkinson's disease, and so he increased neuromelanin. And so he first on the stimulating hormone didn't work, made people worse, maybe. Phenylalanine made people worse. Then he L-DOPA or D-L-DOPA. So he went to D-L-DOPA and they brought the government hospital, built the dose and got the results, and we have a revolutionary drug. That was 1997 that he published this, and 1959 he published the story. So 1967, we're now 150 years from 1817, and of the L-DOPA story, and to this day, L-DOPA is still a therapeutic drug for the motor symptoms of Parkinson's. It doesn't work forever in terms of the disease gets worse and get better. For those who want to know more detail, I wrote and published this year in January. Well, the next big breakthrough was the genetics. It was published again another 180 years after, something like that, after Parkinson's paper. And this was gene discovery. Now, this is a family of large kindred that Roger Duvaucin was looking after in New Jersey. He thought that, well, here we have a family, we can look at the genetics. He got Bill Johnson to work. Couldn't crack the problem, so he turned it over to Nussbaum at NIH, and postdoc Dr. Mihail Palomara was given the assignment. Even when President Clinton annual address to the Congress He mentioned this discovery from NIH that they found this gene for Parkinson's. It turned out the gene was alpha-synuclein, and nobody knew much about alpha-synuclein in those days. They knew it was a system, probably in the nerve terminals, probably bird song. That's all they probably knew at that time. Now today, and since then, a lot of other genes were discovered. I put Park into this one slide. They have to go to the second slide to get one. But then there are a lot of Parkinson's or risk factors for Parkinson's. Park label, and probably the most important gene risk factor was beta-glucocerebrosidase, BA, that. And among other, here's got two very common in India and Multani. I know wrote a paper about that, and people around the world showed that too. And this is Parkinson's yet. And almost every year there's some other gene mentioned cause Parkinsonism of some sort or another. All of these are Parkinson's disease. They may not have Lewy bodies, but they features of Parkinson's. And these are just the point mutation genes. More common genes. GWAS studies. Here's the most recent GWAS study, 1,000 cases, and how many genes? 6 loci were found. So there's probably multiple working together to trigger Parkinson's. It's a common genetic form. Whether the environment's involved, most people think it is. We still don't know quite how, but there are probably some risk factors in the environment. But the genetics probably is the most important. Well, after that first gene that was discovered, some other genes, the one a year later. So altogether, there's 6 different point mutations have been described for this nuclein gene. What made this so important? It's family, very rare. I've probably only saw 1 or 2 in my life, and I've looked, so it was very hard to find these patients. And yet, why is nuclein so important? Well, the first reason it became important was the following year, when they developed antibodies against nuclein because of the Paul Meropoulos study. They studied the brains, and what did they find? They found the Lewy body light up. Here's the typical Lewy body, and for ubiquitin, you get staining in the core, but when you stain for alpha-synuclein, the halo around the dense is what lights up. So a lot of alpha-synuclein is in the Lewy body, and today most people will use alpha-synuclein stains to make Lewy bodies, and that's how they discovered Lewy body dementia and Lewy bodies and multiple system atrophy and alpha-synuclein multiple system atrophy. So the stain became very important. I like this particular slide because I think it's very neat. Japanese workers reported this, and so you can see the halo exactly where that stands for alpha-synuclein. Well, it turned out that initially part 4 was localized, and it turned out when they finally figured it out, it turned out to be also alpha-synuclein, but a triplication of alpha-synuclein, not a mutation. And so there was a duplicate, twice as many normal alpha-synuclein molecules as a normal person would have. And that caused Parkinson's as well. And that was figured out by Andrew Singleton. And the following year, other families were looked at duplication of the normal gene, and it turned out the very largest known family that was ever published in Sweden by Muriel turned out to be a family of both triplications of the alpha-synuclein gene. It turned out also that this is a very severe form of Parkinson's disease when you have excessive amount of normal nuclein. So again, point mutations, staining the body, and excessive normal alpha-synuclein Parkinson's disease, and that's why it's so important, even though families with part 1 are very rare. They mentioned that Lewy body degenerate, dementia, Parkinson's dementia, and of course multiple system atrophy, in which the alpha-synuclein is found in the oligodendrocyte. Matt Farrer showed that the age at onset, his age at onset about 50%, about 50 or just a little under 50 years. By the way, Parkinson's is a more malicious, malignant disease than typical genetic Parkinson's disease. They tend to progress faster. They do respond to L-DOPA, but they get dementia early, they die earlier. It turns out that the triplication ones are even more rare. About a decade sooner, before the age of 40, they have their onset. So this is a bad disease to get the triplication of alpha-synuclein. So what is the function of alpha-synuclein? They knew it was a terminal protein, presynaptic protein, but what the function was still unknown, and it's only been being figured out in the last few years. And among this, I took this from a paper years ago. Basically, alpha-synuclein, which is the monomers soluble in the plasm, they become alpha helix bind to membranes. They do bind to the synaptic vesicle. They're used in vesicle trafficking, vesicle docking, used in fusion with the snare protein, and transmission release. So it plays an important function. So the typical thing, if there's too much monomeric form, can become beta sheets, the oligomers. And it turns out the hypothesis among people today is that it's the oligomers that are the toxic form of alpha-synuclein, and that the oligomers can become fibrillated, forming aggregations, and these are what's found in the Lewy body. And that this hypothesis, the Lewy body is a protective element in the cell. It's not the cause of Parkinson's. It's a disease with the patient is trying to protect, the cells are trying to protect it. They'll take this alpha-synuclein and aggregate it, put it into the Lewy body, get it out of the way. And here you see the helix formation, and more recently there's a tetrameric formation that was a formulation was found. This seems to be stable, and another way to help protect against the monomers going into the oligomers. This hypothesis more or less came from Langley, who at that time was at Mass General Hospital. Until alpha-synuclein can form these oligomers, and the oligomers can go into fibrils of protection. You get too much of this if the synuclein is excessive, if the degradation is not enough, as it turns out, if you get point mutations or excessive genes. So alpha-synuclein could probably be the most important protein in Parkinson's disease, and it's related in almost all cases. About 90 to 95% of all cases of Parkinson's disease have Lewy bodies. There's these small that don't. Most of these are like Parkin and some other genetic form, milder form of the disease. But so important protein, and at least so there's an excessive amount of wild type, the mutations, and it turns out that alpha-synuclein and cytoplasm can bind with dopamine and becomes a modified alpha-synuclein, and that also is a toxic form if that happens. Of course, most of it is not in the cytoplasm, it's in the synaptic vesicle, but sometimes the oligomers bind to those synaptic vesicle membranes and release dopamine into the cytoplasm, and then you get some of it, and that is another toxic form. I will later in my talk about how alpha-synuclein is probably a road protein. We'll come back to that, talk about progression. But also point out that people have developed animals with the deletion of the alpha-synuclein gene. These animals are resistant to various toxins like MPP, excessive dopamine, and so forth for getting cell destruction. So alpha-synuclein looks to be important for all cell destruction of dopamine, whatever it was. So it's a very important protein. Is alpha-synuclein degraded? Well, the typical degradation of all proteins are by the proteasome system, UPS, or the lysosome system. The lysosomes have been known for a long time. The UPS system is a fairly recent discovery. Two Israeli scientists got the Nobel Prize for this. And you can see there are autophagies, chaperone-mediated autophagy, microautophagy, and macroautophagy, and I'll come back to these. But I got this slide from Ron Capito, who's been working on degradation, and I thought this was a very illustrative slide. So you get a protein, it gets misfolded, it has to be gotten, something to be done with this misfolded protein. The first step the cell has to do is to heal it, turn it back into normal protein, and using a certain chaperone, ATP, and that is sometimes successful. If it's not successful, it has to be destroyed. The first step is then the UPS system, that go through the autophagy system. And that's done. Now, I'm taking some slides, particularly a review article by Anna Marie Cuervo and her, at that time, student, Maria Martinez Vicente, and because these diagrams are very nice. So here's the lysosome. By the way, I'm not talking about heterophagy, which is outside substances destroyed inside the cell. Here's microautophagy. We don't need any special trick to get into the lysosome. Macroautophagy, these are usually big structures like mitochondria. They have to have a membrane to go around it. It causes an autophagic vacuole, which then can enter the lysosome and have this degraded. And here's CMA, chaperone-mediated autophagy. And here you see if it doesn't go through ubiquitin, you'll go into lysosome. And so a specific chaperone is developed, attached to it, carries it to the lysosome membrane, the receptor, the LAMP2A protein receptor is attached to it, brings it into the cell, into the lysosome, and for degradation. Now, that was background because it turns out that alpha-synuclein is degraded mostly by autophagy. This was again done by Anna Marie Cuervo and her colleagues, Dave Leonidas Stefanos at Columbia. The normal wild type alpha is normally degraded that way, but it turns out that the mutants, and they only had mutants that time, they studied both, they failed to be degraded. And what happened, they the chaperone, and the chaperone took them to the lysosomal membrane, but they couldn't get in. When attached to the LAMP2A receptor, it couldn't enter, and LAMP2A receptors started getting occupied by this, and the proteins can be degraded. And that is a mechanism of how protein can accumulate in alpha-synuclein disorders, which is what most of the Parkinson's are. So here some alpha-synucleins can be, here's the wild type again, here's phosphorylated, it can't get in, but it doesn't block the LAMP2A receptor. But when you get the mutants, the AT or P and so forth, or the dopamine modified alpha-synuclein, they tie up the receptors, and other cannot be degraded. That is the papers. Now, what about gene mutations? They get LRRK2, GABA mutations, so forth. They get alpha-synuclein. They get Lewy bodies. How do they get Lewy bodies? And what about all these sporadic cases? How do they get Lewy bodies? Well, maybe one of the answers, and this is recent, Cuervo and her colleagues looked at LRRK2 and published this a couple years, 2.5 years ago. Basically, well, I'm not going to spend time to read this. Let me just show you a diagram because it came with an associated editorial, and it has a nice diagram. So here's the LRRK2. Wild-type LRRK2 gets degraded, again, mostly through the autophagy. Some gets degraded through the UPS system, some through macroautophagy. Let's go through LAMP2. What happens here when it binds to the LAMP2 receptor, it triggers oligomerization of the receptor, and now this oligomer cavity and the protein can enter. When you have a mutant, and there's a lot of mutants, probably the most prevalent mutant is the G2019S mutant, which is pathogenic. It binds, but it prevents the oligomerization, and it starts tying up receptors, the LAMP2A receptor, and now alpha-synuclein normally get degraded, can't get degraded. It accumulates, forms oligomers, and you get Parkinson's disease eventually. What about, so I'm just giving you two examples, the LRRK2, DBA, these are the ones that have been worked on. So why do they get pathology of Lewy bodies? And I think the breakthrough on this one came from Dmitry Krank's work at Mass General, and he worked with Ellen Sikorsky at NIH and others. And I'm not going to read this. I want to show you a diagram. Here's a diagram a couple years later by Dansky, a review article in Brain, and it has a nice diagram. I've decided to use this diagram. So here we get glucocerebrosidase, GCase abbreviated, being formed. Here's the endoplasmic reticulum, goes through the Golgi apparatus, and eventually the normal protein enzyme gets carried and it gets localized in the lysosome. It creates its substrate, which is glucosylceramide in the blue. And it turns out, and this work, they were doing that alpha-synuclein, amazingly enough, somehow gets involved with this as well. They tie in with the glucosylceramide, and they get degraded at the same time. Now, when you have the mutation, and as you know, GBA, if you get homozygous, you get gout disease, and if you get heterozygous, in the autosomal dominant form, then you get Parkinson's disease. And what happens here, you get the mutant, you don't get enough active, viable GCase. And so a lot of glucosylceramide accumulates, and the alpha-synuclein can't be degraded. Alpha-synuclein accumulates, forms oligosomes, comes out, the oligomers. You get the oligomers, comes back, and you get this back feedback. So it's a double hit. You get, when the oligomers come back, they interfere with this trafficking of even the normal part of the case. So you're getting double hit, and that could happen even in sporadic Parkinson's disease, it turns out. Sporadic Parkinson's disease has now been shown, and this is by Shapiro's work, they have a reduction in GCase in the brain. Here's the substantia nigra. Here's the those are known carriers. Here's the sporadic PD with the normal GBA gene, and other parts of the well, this is the nigra, and you can see the protein is down as well. And so one idea is, can you somehow transport GCase back into the lysosome? And Shapiro used Ambusol and is now starting a clinical trial. They have Ambusol being used to help sporadic Parkinson's disease or even non-GBA Parkinson patients try to get the normal amount of GCase back into the lysosome. Okay, so that shows you how alpha-synuclein is. I want to make sure that was clear, because now we go to the next step. The next big event happened probably in 2002, 5 years later after the gene was first described, and that was the Brock hypothesis. So Heiko Brock and his colleagues published this paper. Where does it start? He created alpha-synuclein staining antibodies to stain the alpha-synuclein, and normal people were dying, and he found also Louis Neurites, aggregated alpha-synuclein in the axons, and he found that they were starting to appear first factory bulb and the lower brainstem and then over time they were accumulating in places more proximal up the brainstem. And this was the first paper showing out in the nigra in the early stages. It's another place I just mentioned, and eventually he published this hypothesis, and this is I think in 2003. This is a nice picture from this 2004 paper showing the Brock hypothesis. Louis Neurites first accumulated here, and then over time they spread upwards. And so he eventually divided them into 6 stages. Stage 1 is where you get the lower medulla and the factory bulb, and then stage 2, you get the more distal pons, and then stage 3, proximal pons, and the midbrain. The midbrain is involved in stage 3, but the dopamine neurons are not yet affected, so there's pre-symptomatic phase of Parkinson's. We say when those neurons are injured, not functioning well, and eventually dying, you get the symptomatic phase, and then the disease gets worse as the up to the mesocortex next, and then the neocortex last, and that may account for the dementia that occurs later in Parkinson's disease. So this Brock hypothesis has now been more or less looked at. There's another major step in looking at the evolution, and it also account for many of the non-motor symptoms of Parkinson's disease. And this picture from Bill Langston shows the PD features are just the tip of the iceberg. That's the motor manifestation, but they had decreased sense of smell, they have blood function, they have some pain, they have orthostatic hypotension, they have mood changes, they have sleep changes, RBD, and so forth, and eventually dementia can occur. And even before dementia, you get mild cognitive impairment, executive dysfunction occurs, and so forth. And many non-motor symptoms have then been figured out occur years before the motor symptoms of Parkinson's. I've listed some of these here, particularly constipation, hyposmia, REM sleep behavior disorder, and depression can all occur early. Now, some people don't have RBD. Some people get RBD after they get the motor symptoms. It's clear that if you get RBD, it's almost 100% certainty you're going to get Parkinson's disease, MSA, or possibly Alzheimer's, most likely one of the Parkinson's syndrome. And then Brock and his group put together their best guess. A clinical onset, we'll start with that. ONER stage 1, unilateral, stage 2, up to stage 5, wheelchair bound. And you can see it starts not in the beginning disease, but maybe 20 years after when Brock stage 1 occurs, and with hypoxia, constipation, bladder disorder, stage 2, sleep disorders, depression. Stage 3, you're not starting to get into stage 4, get Parkinson's symptoms and so forth, all the way up. And this is possibly a very effective model, and he talks about where the involvement is in the alpha-synuclein aggregations. Okay, so now I don't want to leave you think that all Parkinson's disease is alpha-synuclein, or that's the only source. I mean, there are other factors. There's certainly oxidative stress going on, mitochondrial dysfunction, that protein accumulation we just talked about. There's inflammation. Some of this is going to be talked about by Etienne Hirsch. But I just want to show you this slide. All interplay with each other. These seem to reflect each other. Some are more mitochondrial, like Park, and less protein accumulation for that disease. But you see the genes are playing a role, maybe the environment is playing a role. There's certainly mono cells are much more susceptible to oxidative stress. The monoamines are, of course, oxidized to oxidative compounds. The next break step, I would say, in breakthrough is the rogue protein hypothesis. Now we know now we have the broad hypothesis, and this new hypothesis is that alpha-synuclein is a bad protein. It's a rogue protein. And this all started with this publication. 3 papers back-to-back, Nature Medicine 2008. These were pathology reports of people who had received fetal tissue transplants more than a decade earlier, 11, 14, 12 years before when they died. Later on, some of those cells, those transplanted cells, surviving those neurons from the fetus, now had alpha-synuclein inclusions that looked like antibodies. There were a minority of the cells, and not every brain had them, but some brains did, and that raised this possibility. This is from Patrick Brunn and his group, and here Porter and his colleagues. They did some open-label fetal tissue transplants. This is from Canada. They looked in their tissue. They did not see these changes in their tissues. I would say this is a minority of the cells that get change. But this is from that paper. These are the inclusions same with alpha-synuclein, for alpha-synuclein. Well, that led immediately to researchers working on this. How does this happen? And this first paper came out a year later. This is tissue cultures, and they show that alpha-synuclein can be transmitted from neuron to neuron tissue culture. That led them, PNAS, the journal, to get an editorial, and they asked Prusiner, who teamed up with Born-Olano, and they suggested that maybe alpha-synuclein is a rogue protein, a prion-like protein. Like the prion disease, all people have normal prion protein in our brains, but sometimes prion protein changes to, well, call it a rogue protein, we call it a prion, and it makes normal protein a prion, and this accumulates. You get rods, you get plaques, amyloid, and you get Lewy bodies in similar fashion. This is the prion hypothesis Prusiner and Olano have been using. We'll come back. That's the right hypothesis. Some people will disagree with it. But then other work. Here's another work. This again in culture. this is where they took synthetic alpha-synuclein fibrils. This is a Virginia Lee's group. Alpha-synuclein fibrils injected into the brains of the rodents, and they found cell death. And they injected into the, no, this is cell culture. This is cell culture, sorry, that's the next paper. This is cell culture. They found cell death and Lewy body type relations of alpha-synuclein. And so I'm going to read this. And then a couple years later, they did it in mice. And this is the one I'm saying. They injected these synthetic fibrils, alpha-synuclein fibrils, in a couple areas of the brain, and they found, for example, they injected in the stream, they found Lewy body formation death in the nigra, and they found it in another area of the brain as well. So that this says Lewy body accumulates progressive loss of neurons even when you inject it into the striatum, you get it in the nigra, but not in the next area. So, and then this next paper was showing that alpha-synuclein can leave the cell, go extracellular, and come back to cells, and it can be transmitted that way as well. So there's exocytosis of alpha-synuclein, and it can be endocytosis into other cells, and you can get not only neuron-to-neuron that way, but by the extracellular route has been shown. This paper from Japan, again, looks at the spread, and I think, again, what they were showing, again, it's really transsynaptic spread. When you inject it, it's where those connecting pathways inject into the olfactory bulb area, you get those pathways connected with the olfactory bulb, and so forth. So it is spreading neuronally. And then Lee, one of the natural things to do, can you stop this? She developed antibodies to alpha-synuclein, and after injecting the synthetic alpha-synuclein fibrils, injected the antibody, and prevented the spread. So stop the propagation. So by tying up the alpha-synuclein, you can do it. Now, what will be important is not to do it immediately, injection of the antibody immediately, but wait a couple of weeks, wait a month, and see if you can reverse the damage. Is what we need for, of course, treating people with Parkinson's disease. And so that's what this paper showed. And then most recently, I shouldn't say the most recent, it was just a year ago now, Miquel Vela in Spain, in Barcelona, who had worked at Serge Zaborski's lab at Columbia and then went back and got his own lab in Barcelona, he took extracts of Lewy bodies from people who died of Parkinson's disease and injected them into of mice and monkeys, like the prion study. Colin Masters is right here in the front row, heating this all up probably. Studies that they were interested in, because they injected brain tissue. They didn't inject Lewy bodies, they injected brain tissue. They injected Lewy body extract, and they got degeneration. They enriched and they found both mice and monkeys, intranigral or intrastriatal inoculations cause progressive nigrostriatal degeneration, starting at the dopamine cells, not in the cell bodies. When they received non-Lewy body actions from the same patients, they did not get degeneration. And why is the first site of Lewy aggregation, Lewy neurites formed in the distal medulla, the dorsal motor nucleus, the vagus, olfactory bulb? Is this coming in the environment? Is this something we're eating? Is it something we're breathing? This is all speculations. It's not clear. In this particular, this is a speculation paper by Pat Brendan and Kathleen Shannon, colleague, and about possibly the microbiome in our guts is allowing entry of alpha-synuclein, and that if you have protective factors like perhaps smoking and coffee, change the microbiota, and you don't maybe, or some other factors are making it worse, inflammatory factors, alpha-synuclein enter, it enters the nerve terminals of the vagus nerve, it goes up the vagus nerve into the medulla, and from there it spreads. And so that's good timing. So what I said at the beginning, people are willing to tolerate the early symptoms to stop the spread. Can things like antibody treatments, immunotherapy, stop the spread? already there are pharmaceutical companies that have developed antibodies, and much like we heard the first lecture from John Morris about antibody treatment for people with Alzheimer's disease, this treatment is going to be starting for Parkinson's disease, and we will see what happens. And I know one company already is starting to develop the protocols for treatment. I want to end by saying that most of the protein, most of the gene mutations eventually cause accumulation of alpha-synuclein, Lewy bodies, and you get the spread of the disease. With typical Lewy body Parkinson's, whether it's through a mutation or whether it's sporadic, once you get alpha-synuclein, you get this problem. There's a few types of genetic mutations that don't do this. I've already mentioned Parkin, but one seems to be the same, DJ1, BX07. They affect the recessive genes. They tend to affect mitochondria. They do not form Lewy bodies. At autopsy, they don't have Lewy bodies, by and large. And they don't have dementia. They don't have REM sleep behavior disorder. They don't have the depressions, and so forth. But they do get all the motor symptoms. They get all the motor complications from levodopa, and they can even get some freezing of gait. So they do get motor Parkinson, but they don't get all the non-motor. My last slide is to tell all of you about the World Parkinson Congress. It's going to be next September in Portland, Oregon. This will be the fourth one. There have been 3 up to now. All been successful. But what's so important about it is where the neuroscientists, the Parkinson clinician, and the patients and their families will all meet together. And about 1/3 of each group of all the population represents each group. And there's plenary sessions and there are parallel sessions. People gather together talking to each other, and I want all of you to come. So try to get some funds, and there are funds for junior investigators. There are grants. They haven't been announced yet because it's still about a year away. But put in your calendar, whatever the date is, September 20th to the 23rd, 2016 in Portland, Oregon, and hope to see you there. And thank you very much for your attention. I'll be glad to answer questions.
Thanks for a wonderful overview of disease, and I guess since we now know a lot more about Parkinson's disease, maybe this will probably be instructive for understanding the diseases that we know less about. We have time for, I think, 1 or 2 questions.
I should say it's a wonderful presentation. The overview of Parkinson's research, very methodical and very logically progressive presentation. Quite impressive. Thank you, sir. And I would like to ask you a couple of points I wanted to make, but I would go for the most important one for me. So now that this prion hypothesis is being emerging and being extrapolated to many of the neurodegenerations, say Alzheimer's, Huntington's, now as you mentioned the Parkinson's area, whatnot, MSA, multiple system atrophy. So are we going to get a common solution by studying this transcranial propagation, or do you think it's really imminent for neurons to communicate transsynaptically many proteins, and this is just a part of it? And also whether this contribution of transcranial propagation is a major thing in all these pathologies, and to how extent?
How is it? Well, I mean, why should alpha-synuclein be a rogue protein? I mean, why does prion protein become a rogue protein? I mean, something happens about it. I think that mechanism still has to be understood, and maybe Colin has some answers about that one. But I think there is spread. I know best about Parkinson's disease and about the alpha-synuclein and the Brock hypothesis. We can see the spread. We can see new symptoms in Parkinson's developing. I'm not an expert in Alzheimer's. I don't know what new things develop once you get, of course the disease gets worse, we know memory gets worse and so forth, but is it really spreading or is it just getting worse where it's already present? So I don't know about spread in these other diseases. And whether it's through a rogue protein mechanism or something else, the degenerative process just gets worse.
I mean,
and the rogue protein hypothesis has not been proven, it's just a hypothesis still.
But that was a quite good evidence of rogue protein hypothesis anyway. Quite a few papers you have mentioned. And like what is the level of contribution to the pathology was one question. Whether it is so rewarding to study in all the pathologies, this transneural propagation. And anatomically non-contiguous areas are not explainable with transneural propagation. So it might just one of the factors out of many things.
Well, I think imaging, I must say, a big advance in Alzheimer's, which we don't have in Parkinson's, is the fact that they can image amyloid already. It's not that alpha-synuclein, they're not trying to find a stain to localize alpha-synuclein. That would be terrific. And you might be able to measure progression of disease, the spread of disease, if you have a good imaging device. Right now, we don't have that for Parkinson's disease. That is certainly on people's minds and the people working on it. And I suspect someday we will have it. And that might be an early biomarker, and if you have a treatment, you might start earlier, even the presynaptic phase of Parkinson's disease, and you can